Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, the Health, Medicine, and Bioscience edition. My job here is to find the top people in their fields. This could be 1 in 500 people, 1 in 1,000, but they're usually really good at what they do. I want to ask them questions they haven't been asked before, hopefully, and get some great information for you, the listener, so that you learn something and maybe it improves your health and your life or your family's life. That's the goal here. So today I have Paula Barreras. She's a postdoc at Johns Hopkins and working in neurology on brain organoids. These are approximations of the brain. They may be really good approximations or really narrow approximations, but scientists can test various drugs on them and protocols to see how a real brain might work, and then you don't have to use an animal, hopefully. So that's my understanding of organoids. But Paula, thanks for coming. How are you doing?
Doing well. Thanks, Richard, for inviting me.
And I hope my organoid brain was able to speak about brain organoids in the right way. I mean, is that correct what I said?
Yeah, so this is a very interesting new technology. It's basically a small sphere of human cells that we manage to convert into neurons and glial cells, which are the supporting cells for the neurons, in co-culture in vitro, which is notoriously hard to achieve. Like you said, most of the studies are based on animal models, and animals are not humans, not genetically or physiologically. And, that creates a big issue of studies failing when translating to human studies from animal studies. And we believe that the use of technologies that are human-based can help bridge, that barrier in drug screening and other studies.
So, I know we can't make a whole brain or a whole organ, but so what functions of the brain are you trying to approximate? What kind of cells and, again, what kind of structure?
Right, so so we're trying to first achieve the presence of mature neurons and glial cells, which is, different from just a ball of stem cells. And we are trying to approximate the interactions between these cells as close as possible to normal human adult brains. So these mini brains or brain organoids have actually a few features of mature human brains, including synapses, meaning neurons talking to each other and interacting in a functional way. There's evidence of early myelination. That's oligodendrocytes, which are the cells that make the wrapping around axons. We're seeing those mature cells present in our culture, and they're starting to wrap around the axons, which is something novel of this model. And there's also spontaneous electrical activity.
No, that's amazing. What kind of medical conditions do you hope to approximate or test with the organoids?
Well, the great thing about this model is that so the way it's made is starting with skin cells of a donor. And you could imagine that you can use any donor, either a healthy volunteer or a patient with a particular disease. So in theory, you could use this model to do personalized testing for many different brain conditions. We've used healthy donors to produce healthy brain organoids and expose them to different viruses, for example, to test for susceptibility of different types of brain cells to infections and understand more about these type of infections. We used it, for example, with the Zika virus when the outbreak happened in 2016. But the other thing that we can use them for is to study particular brain disorders by using or producing organoids from patients. In particular, since this model has myelin, which is very hard to have in culture, you could think about using it for demyelinating disorders such as multiple sclerosis.
So That's weird. Using skin cells, I guess you have to walk them back development-wise to induce pluripotency in them, and then guide them on the pathway down and become brain cells, right?
Yeah, that's correct. So we start with adult human skin cells. Those are called fibroblasts, and then we induce them back to be stem cells. And from that stage, we induce them to become neural progenitor cells, which later develop into neurons, astrocytes, and oligodendrocytes, which are the main cell types in the human brain.
That must have taken a lot of time and effort to figure out how to first induce pluripotency and then, walk them down the pathways they need to go to create all those kinds of cells. But, there's a lot of people working on this. So
it did. So we're not the first group that manages to produce pluripotent cells. Fortunately, there's a lot of very smart people working in how to use these technologies. But I do think we're one of the first, if not the first, to manage to have co-culture of these adult phenotype brain cells with functional features like this. But it did take a lot of trial and error and long nights at the lab.
So where are you at right now with the research? Are you still trying to tune the organoids so that they will approximate the functions you want, or they're good enough and now you want to start testing the effects of different diseases on, for instance, myelination or other things?
Well, we think they're good enough now. The first main part of the research was try to not only produce them, but make them standardized such that every time you would produce them, they would be similar to each other. And that phase is done. We think we are able to make a reproducible model and make them similar to each other each time. Then the next phase was to characterize, which cell types we have, how mature are they over time, what's the optimal time to, culture them. And we think that's optimized now. And right now we're starting to use them for testing different molecules or different experiments. One of them is, for example, JC virus, which is a virus that can cause severe neurological disease in the immunocompromised, in people with HIV or taking immunosuppressant drugs. So we have started some experiments to expose the brain organoids to this virus and have gotten some interest.
What is the blood-brain barrier composed of? And do you think that might be necessary to really approximate the function of this organoid? What if you were able to culture a barrier and then you let the virus loose on the outside of the barrier or whatever it was, and it had to migrate through it, and then maybe it would approximate what happens in the brain better? I don't know.
Yeah, that's a great question. And it's probably one of the main limitations of this type of model. The model doesn't have blood vessels, and therefore we cannot have a brain-blood barrier in that sense. So the blood-brain barrier, like you sort of said, is the barrier between the blood vessels, and the brain. And there's a lot of cell types and interactions between the blood vessel wall and the astrocytes, which is the main cell type that helps form that barrier. And we don't really have that in this model. So by putting the virus directly, we're assuming or recreating the scenario where the virus already went through the brain-blood barrier. There's been some experiments that we've tried to approximate what a blood-brain barrier would look like by using endothelial membranes and putting the brain organoids in one side and the toxins in the other side. So there are ways to get around it, but our model doesn't have blood-brain barrier as such.
What about the different parts of the brain? I mean, it'll be really hard, I guess, to even create mini versions of the medulla, the cortex, everything. I don't know, I guess, is the model sophisticated enough where you'll learn plenty from it at this stage? And, is there a plan to really make truly a mini brain with different areas?
Yeah, well, those, like you said, that's very hard to make. And I think the models that has been attempted by other groups and, with limited success. And the reason that's the case is because as the larger an in vitro model becomes, just the size, because there's no blood vessels, there's no nutrients that can reach the center of the sphere or the center of the model, and then it starts causing cell death in the middle. And then as the more complex a model is, the more variability there is between each individual organoid. So our model is more simple in that sense. It's an aggregate of the 3 main cell types, and it's small, it's 300 microns. But the good thing is that it's always the same. So we cannot use it to say study specifically what would happen in a region of the brain, but we can use it to study specifically what would happen in a particular cell type or with the interaction between, let's say, astrocytes and neurons in a particular viral infection or toxin exposure. So it serves a little bit of a different function, but we think that's actually better if you want to create reproducible, scientific parameters that you can measure.
Interesting. Has anyone in the organoid field been able to grow blood vessels so they can make, denser or more complicated structures, or is that just a holdback on anyone working in organoids?
So there's some groups producing blood vessels, and that's like its own project. I don't think, to my knowledge, that there's a brain organoid with blood vessels incorporated in it. But that's certainly a next step that I think is worth exploring, because it's a big limitation to grow them in larger sizes.
Okay. And then, it's one thing that came to mind. I know in a lot of cell types put out extracellular vesicles, lots of them. But in the brain, I'm sure a lot of the cells communicate through, neurologically. I wonder if they have much of a need for EVs, and if they put them out, and is that a part of your model at all? Have you seen any of them come out of the organoid, out of the cells?
So if I've seen vesicles come out of the organoids?
Yeah, if they're putting out any extracellular vesicles, the organoid cells.
Yeah, so we saw a few things. We saw that there are synaptic structures, and when there's synaptic communication between neurons, there are vesicles of neurotransmitters that are produced, and we were able to measure markers of such neurotransmission in the model. So we know that there is at least that type of vesicle being produced. When we did electron microscopy, we saw evidence of small vesicles inside the cells being produced. So there is some of that. We haven't really measured that as an outcome, like as an endpoint in the extracellular fluid. But for example, when we were testing for infection, we were able to measure new viral production by the organoid, which is presumably, through excretion of vesicles containing virus. So
any interesting behaviors you've seen from the organoids you make? Or, you're testing, like you said, certain viruses on them. What any experimental results that are really unusual or cool?
Yeah, I think one of the coolest things of this model is the presence of myelin. And, to understand how important that is you have to understand that myelin is produced by oligodendrocytes, and they are very hard to culture. And because of that, most studies in diseases like multiple sclerosis are based in mice, and a lot of the trials fail and whatnot. And that's probably because the biology is not the same. This model has mature-looking oligodendrocytes, which is rare, and it's actually a surprising finding that we were not expecting to see. And then when we saw that these oligodendrocyte cells are actually wrapping the axons, we were very happy and very surprised about that, because that's usually not what you achieve in culture. I think we're seeing that because the presence of astrocytes in the same culture enhances the normal, it approximates more the normal biology of the human brain, and probably there's signaling that you wouldn't have in let's say a culture just with oligodendrocyte precursors. So this presence of myelin was very exciting for us, and we can quantify it in a standardized way. And then we saw that the older the organoid gets, the more myelin it's produced. I think that's a big deal for us, because now we can start thinking about testing agents for remyelination, for example, which we haven't done yet, but it's sort of like the future of the multiple sclerosis field, I think. For viruses, we were able to infect these oligodendrocyte cells with JC virus, and the JC virus is a very tricky virus because it can only infect human cells. And because of that, there's no animal model to date available to study that disorder, and therefore no effective treatments, because there's really no model to test the treatment in. So now that we saw that we're actually able to achieve infection in that model, that's very exciting, and that, have us thinking about testing different chemicals, thinking out new treatments for that disorder.
Okay. I'm not sure what to ask you about this. Well, in terms of protocols, I know that mouse models are like the gold standard. In general, when do you think organoids will be part of the the clinical trial process? When will they be used more than animal models, let's say?
Well, I think that's the dream in a way. So animal models have a lot of limitations, like I've said already, but also some ethical issues with, animal treatment and cruelty. And if we are able to prove that you can get not only good enough data, but even better data based on having human cells, in a model than animal data, then I think that they will sort of turn the corner and say, okay, let's use more relevant human models for clinical trials. I don't think we're quite there yet because this technology is relatively new, and a lot of the drug developing companies are very familiar with animal models and not so much with this type of technology. But I think there's more and more interest from drug developing companies in this type of technology. I imagine in the next 10 years this will be used more and more, and maybe at that stage we'll change gears and start using this type of technology instead of animal models.
Yeah, I figured because you're in the field, I just wondered what an update was. Is it still way far in the future, or does it seem to be getting close?
I think it's getting closer and closer. There's, depending of the use, I see one thing's closer than others. For example, using this model to evaluate toxicity of different substances, I think that's closer in the future than using it, for example, to study diseases like multiple sclerosis. Using it to test compounds as treatment for viral infections, I think that's very plausible and near in the future. It's relatively easy to do. We can produce 800 of these organoids at a time.
Oh, wow.
So we can produce many of them and treat them at the same time, put them in different wells and expose them to many different substances. And that makes it way cheaper than doing that with animal models that take longer to be produced, and they're more expensive. So I think the drug testing companies will be interested in this type of model just because you can massively produce it, it's cheaper, and it's presumably more accurate depending on what you want to measure.
So when you do your experiments, are you able to do you do 100 organoids at a time? Are they really cheap to make and easy, or are they still difficult?
I think they're cheap and easy to make. We make, let's say, 800, and when we test, I would say I use something like 80 in an experiment. With a standard would be, let's say you're testing 3 different experimental conditions, and you put 30 or 20 in each well and expose them to that, and you don't need that many. You with just one organoid you have a lot of information, but for reproducibility of your experiments you want to test up to this 5 of them per condition. So it's really easy, to have enough material to reproduce experiments and test multiple conditions. And that's not true for, let's say, mice, because they're harder to work with.
So what does that allow you to do in your experimentation? I guess you're able to, it's like having, a whole bunch of animal models in a sense. I mean, you're able to, test many at one time.
Right, you can test many at one time, and I think importantly, many different conditions at one time. So you can put, a dish with 90 wells and put 5 of these organoids in each well and test 90 different conditions in one experiment, which is really hard to do with, larger, bulkier, models like a mouse.
Okay. All right. So what other experiments are you doing? What are you trying to figure out right now with organoids?
Well, right now we're focusing a lot on the JC virus infection. Another thing we were trying to do is working with HIV, because there's a condition so in people that have long-standing HIV, we're starting to see cognitive decline, basically dementia, that is believed to be caused directly by the virus. And some studies have suggested that that's the virus infecting some of the glial cells, and the mechanisms by which that happens are not well understood. So we are interested in looking into that, exposing these organoids to HIV and sort of studying the susceptibility of these different cells to the virus, and see if there's anything we can do to, stop the spread. Because this happens, this cognitive decline happens despite people being on adequate therapy for the HIV. So the mechanism is felt to be different and is not understood. So that's our use of this model. You can use it to not only test compounds or test infections, but to try to understand the mechanism of disease, because you can measure gene expression, cell-to-cell interactions, susceptibility of different cells. So we're looking into that for the HIV infection as well.
Okay. Well, very good. What's the best way for people to find out more? And maybe, I don't know if they could see some of the organoids, if you have any pictures, but how can they find out more from here?
Yeah, I think we have a couple papers on the model that have pictures. I think so. This model was done in collaboration with the School of Public Health group at Johns Hopkins, and that's David Pamis and Helena Hochberg, and they have in their website a video with like a YouTube link to a little bit of how the process is made. So that would be a good way for people to find out. But hopefully we'll be putting out more information and more papers that people can access.
All right, well, very good. Well, Paula, thank you for coming. The organoid field is, like I said, it's amazing. It's really cool. And, the brain is probably the most interesting part of it. So that's great. What's the best way for people to, again, find out more? Is it a URL of a website where your research is? How can they do it?
Yeah, I think if you go to the website of the School of Public Health Lab, they have a good video about it. And I think if you type mini brain Hopkins, then you get very fast to the website that we have there with some basic information. There's a link to a video that people can access.
Well, if any person comes up when you put in mini brain, that would be an insult to them, but this is not what that is.
Right.
Well, very good. Well, Paula, thank you for coming. It's been a very cool call. I appreciate it.
All right. Thank you for inviting me.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.